Welcome. Today we have Brian Cleary, who's joining us from Boston. He's an assistant professor in the Faculty of Computing and Data Sciences and the Department of Biomedical Engineering and Department of Biology, and is co-faculty member of the program in Bioinformatics and the Biological Design Center at Boston University. Professor Cleary received his PhD in computational systems biology from MIT and was an independent Broad Fellow at the Broad Institute before going to BU. So today, Brian will introduce to us his work in compressed sensing. A lot of that work was first done in bulk, then on kind of spatial, and a lot on the perturb measurements. So welcome, Brian.
Thank you. Let me get hooked up here. Nope, swing and a miss. There we go. Okay, good. Can hear and everything? Right. Thanks much for the invitation. Been very much enjoying my visit so far. Excited to tell you a little bit about a work that I was, somewhat upset to be reminded that this work dates back to maybe 2017 or 2018 or something. But anyway, started in my PhD project, and it's a funny project where I've been presenting on the work since sort of the preconception stages of the work and at basically every intermediate stage in between. So it feels like, old news, and yet there's no thing that exists, but happy to say that we're actually submitting a paper next week. And so, I can move on with my life. No, but seriously, this is very fun, sort of central to a lot of different work that we're going to be doing in the lab. And I think is one implementation of what is a very general framework that hopefully has applications for many of your work. Just a quick note about where I am. There's this wacky new building that exists on the Boston skyline on the Boston University campus. So I'm primarily appointed in what's called the Faculty of Computing and Data Sciences, which is effectively like a college level thing at Boston University. But it's meant to be the sort of thing where there are folks in there working on data sciences, interfacing with departments all over the university in history and the law and sociology and science and engineering and everything else. So it's kind of fun, to have colleagues who sit next door and working on data science and the law and so on. But anyway, the point of that is both that there's this interesting new thing that you could check out if you're interested in going to Boston, but also that sort of the context of the work that I do is very much not just using data science or algorithms as applied tools in biological contexts, but really using them fundamentally as a lens on how we might solve biological problems, hence an algorithmic lens on functional genomics there in the title. And hopefully it'll become a bit more clear, at least throughout the introduction, what I mean by that. A quick bit of overview on the work that we do in the lab. In terms of sort of biological systems, or really scales, we tend to focus on 3 different scales. The first is that we're interested in interrogating sort of intracellular circuits. For example, we might like to know for a particular cell type, we want to know how it responds to a certain extracellular input or stimulus. And we very often do this by developing some way to measure in a high content fashion the state of a cell, and then we'll make different perturbations under stimulus and see how the state of the cell changes. So we couple together usually high throughput perturbations with high content measurements to try and interrogate these intracellular circuits. We're then interested in the same sort of questions, but at a slightly larger scale. So instead of intracellularly, we're interested in the same sorts of things intercellularly. So if I perturb one cell, how does the state of a neighboring cell change? And then, theoretically, we're interested in the same sorts of questions at larger scales. So moving from a single cell to a microenvironment to entire large tissue volumes or an entire organ. And there, we're in the very early stages. I don't even know what I mean to say that we want to do the same thing there. Roughly speaking, I want to perturb one microenvironment and see how a neighboring microenvironment changes. But that's pretty much the extent of what I know I mean about that. So really what's happening at that stage right now is developing the basic technology to measure the state of microenvironments in high throughput, or really measure over large tissue volumes in high throughput. which isn't particularly well captured by the words on the slide there. So listen to my words and not the printed words instead. Okay, so in the context of those three different biological scales, we have three primary branches of work. One is just basic technology development. So where tools do not yet exist to measure the state of the system, either intracellularly, intercellularly, or at large volumes, we might develop those tools, and similarly for making perturbations and coupling the two things together. Usually we're interested in genetic perturbations coupled together with high content measurements of the expression of the system. The second thing that we do is where technology already exists to make high content measurements and make high throughput perturbations, but where these technologies cannot be scaled up to comprehensively profile a system in a way that we might ideally like to do, what we do is apply what you can think of as the mathematical foundations of how we learn about very high dimensional problems that are structured in sort of a latent way using certain classes of algorithms and very small amounts of data as inputs. So that was like a 90-mile-an-hour mouthful of stuff. It's going to be the focus of the rest of the talk. So I'll get into a little bit more about what that means. The third major branch of work that we do is kind of moving beyond snapshots of the state of a system to looking at the dynamics, short-term dynamics, either intracellularly or intercellularly. And in particular, we're interested in interrogating how perturbations affect not just a snapshot of the system, but how a system is evolving locally over a short period of time. Okay, so as I said, today we're going to talk about this particular biological scale, interrogating intracellular circuits, and our work that, as I said, has been sort of accumulation of stuff done over several years now, scaling up existing technologies with what we call compressed experiments. So I'm going to start off by giving you kind of a broad mathematical foundation that underlies a lot of different work that we do in the lab. As I mentioned, what I'm going to talk about today is sort of one specific implementation of this very broad idea of taking an algorithmic lens on biological experimental design to increase throughput, hopefully in in ways that generate exponential gains in efficiency. All right, so broadly speaking, in the different settings that we look at in the lab, there are limitations to throughput that are very often based on what seems to naively be entirely sensible. Like if I have a bunch of variables that I would like to measure, I should measure every one of those variables. That seems like such a dumb statement that it doesn't even make sense. Or if I have a lot of different things I would like to perturb and know what the outcome is, I should perform all of those perturbations. Also, it seems like, well, yes, that is so obviously true that why are you even saying that out loud? But it turns out to not actually be necessary. If you want to know sort of the expression level of every gene, you don't have to measure every single gene. And there are good mathematical formulations that tell us when that's true and how we should go about it. And the same thing is true for perturbations. If you want to know the outcome to a large panel of perturbations, you don't actually have to perturb every single thing and measure the outcome individually. But you can do a much smaller number of experiments. And we have theoretical understanding to tell us the limits of how much data we actually need to collect. So all of that is to say, what is the second sentence here, that there are certain limitations in experimental biology that will never be overcome by like building a better box. Okay, it doesn't matter how Ultima Genomics is or whatever is the name of that company or any other company. There are certain problems that cannot be solved by taking this conceptual approach that already exists and just like running it in a box that runs faster or generates more stuff for the same amount of dollar. This is particularly true in combinatorial problems, but is true in other realms as well. For example, you know, people are increasingly trying to measure mRNA molecules in space, and measuring every single mRNA molecule across an entire brain. You know, how many molecules are there in an organ? A lot. We don't need to measure them all. That's madness. All right, so what are some of the mathematical foundations of how we think about this problem? We're going to go way back to mathematical foundations of high dimensional data and thinking about similarity of points in high dimensional data to a result, something called the Johnson-Lindenstrauss lemma that underlies a lot of the success of sort of low dimensional representations of data in any sort of realm. Okay, so this is a very powerful result that says that for any set of n points in high dimensional space, any data, okay, whatever it is, doesn't matter where it came from, there always exists a low dimensional embedding requiring only about log n dimensions that preserves all the pairwise distances between the points with minimal distortion. So for example, in biological problems, we very often collect very, very high dimensional data, and we're very proud of ourselves that we spent lots of money to collect high dimensional data. And then we shoot it down to 2 dimensions or whatever, or we do clustering, or we do something that depends simply on the similarity between samples. So for example, clustering things into cell types or something like that. And for these questions of similarity, it's just interesting to note that you don't necessarily need all that high dimensional data. So you don't need 20,000 dimensional data point to accurately cluster stuff, that there always exists, no matter where the data came from, a representation of those data in low dimensions that preserves that sample to sample similarity. And so what this inspires is kind of, as I said, a reimagining of the typical approach to experimental design and computational analysis, where the normal method, as I said, is we collect all this high dimensional data, and then we project it down and do various bits of computational inference in low dimensions. And if our inferences that we're doing always only depend on these low dimensional projections, instead, what if we could directly acquire the data in a low dimensional representation and perform whatever computational inference that we're going to do there? So instead of acquiring like n different measurements or performing n different perturbations, if we're doing some analysis that depends on the similarity of all those different variables, then we're just going to project it down anyway. So I could just save myself all that work in the lab and only collect something that corresponds to this low dimensional representation, and the whole experiment might be like exponentially cheaper. Okay, so that's the very broad sort of underlying mathematical foundation. This can be extended through something called compressed sensing that says that if you have, so this initial result that high dimensional data in general can be projected into low dimensions while preserving the geometry, can be extended with something called compressed sensing to say that if you have prior knowledge of structure that exists in your high dimensional data, which can be defined in a variety of ways, then in fact, from these low dimensional representations, you could reconstruct the full high dimensional signal. So even if you, for some reason, do care about the individual outcomes to n different things or the individual expression level of n different things, you don't need to just do your computational inference in low dimensions. You could reconstruct that full high dimensional signal from the low dimensional data. So that's basically what we're doing in the lab, is that we can learn, we're taking this insight that we can learn about highly structured systems far more efficiently than naively thought. And we're sort of exploring in kind of a fun way how the organization of biological systems relates to sort of the fundamental limits of biological experimentation through this sort of algorithmic lens. Again, where the difference here, this is just propaganda, it doesn't mean anything. It's purely to advance my career with some term. But unlike using algorithms just as a tool, okay, that we run the experiments however we run them, and then we use algorithms as a tool, here we're kind of using our understanding of how the algorithms work to rethink how we generate the data in the first place, and with an idea of, we're going to apply certain classes of algorithms to analyze these data, and here's a way to design the experiment that maximally sort of makes use of the power of those classes of algorithms. All right, so now what are we actually doing today? We're talking about perturb-seq, theoretically, eventually. So here's the basic idea of how we're going to, or sort of the relative scaling of what we can think of as conventional screens versus compressed screens. Okay, so in a conventional screen, as I said, we might be interested in whatever context in understanding the outcome of n different perturbations. And for now, let's think of just kind of a one-dimensional outcome to a perturbation, like survival or fitness or the expression of a single gene or something. So a normal person, thinking I want to study n different perturbations, will generate something like on the order of n different samples, where the sort of big O notation here captures whatever constant factor relates to the number of replicates that you need to do in your experiment. And then from those n different samples, you're gonna learn some effects, which perturbations produced an effect. Let's say there was like k non-zero effects. In a compressed screen, instead, we still have n different perturbations that we'd like to learn about, but we're going to generate a much smaller number of what we can think of as composite samples. And the number of composite samples that we need to generate is now motivated by these theories of compressed sensing, also related to things like pooled testing, if you're familiar with that, on the order of O of k log n, where again, k is somehow related to the sparsity of the result. Okay, so instead of having to perform n different things, now the number of experiments that we need to perform should scale as the log of n. Okay, so we have an exponential gain in efficiency. And then we have a linear scaling based on the number of non-zero parameters that we're trying to infer, which we don't necessarily know in advance, but very often we know that many perturbations don't produce a significant effect. So we have some prior notion that there's going to be sparsity in the outcome, even if we don't know exactly how many non-zero things they're going to be. So that has already been implemented in quite a variety of ways. As I said, with COVID, people are now familiar with pooled testing. That is sort of one, it's not exactly this, but it's related to sort of the mathematical foundations of why pooled testing works are essentially the same as what's going on here. I should note that, in conventional screens, there's a whole lot of different inference methods that work. As I mentioned here, this sort of design where instead of n samples, we're going to have k log n samples only works if we're assuming we're going to analyze the data with certain class of algorithms in particular that take advantage of sort of the sparsity in the result here. So if you're familiar with like lasso, for example, that's one that could work well. But anyway, again, related to pooled testing, just to skip ahead to the end, you can think of what we're doing in the experiments that I'm about to show as effectively like pooled testing inside single cells for the activity of a variety of molecular programs. Okay, so that's not exactly what we're doing, but it's close enough, if that's helpful to think about it that way. All right, so this was again for kind of a one-dimensional phenotype. Now we're going to be doing these perturb-seq experiments where we want to infer the effect of genetic perturbations on not just a one-dimensional outcome, but on a high-dimensional outcome, in particular, the expression of every gene in the genome. And so the algorithms and the scaling are slightly different. I'll get into the details in a little bit. But now we need something, instead of performing n different perturbations, we need something like log n times r plus q. Okay, and what are these things? R is something like the rank of a matrix of the data that we observe. And q is related again to one of these non-zero or sparsity type parameters. Specifically, q would be like the number of non-zero effects per sort of perturbation module. I'll come back to that in a little bit. All right, so that's the general scheme. And again, the idea here is that this is a very broadly applicable framework. We're doing it in the context of genetic perturbations to study sort of transcriptional regulatory circuits. But this could be broadly applied to sort of any screen, generally speaking, with some assumptions that I'm happy to talk about later. Okay, so we're doing it in the context of these pooled CRISPR screens with a single cell RNA-seq readout. For those of you who aren't familiar, here's how a conventional perturb-seq screen works. We're going to use CRISPR-based genetic perturbations. So we have a guide RNA library targeting different genes that we're interested in perturbing. We introduce those into cells, usually at low MOI, so each cell receives one perturbation. In our case, we're using single cell microfluidic-based sequencing. So we load these cells into droplets, again, at generally low concentrations, such that each droplet ends up with a single cell. And then that cell is lysed, all the RNA gets dumped out, and we end up sequencing it. So we observe that sort of output of what we observe from these experiments is in each droplet that we observed, we get sort of a count matrix of how much every gene was expressed within that droplet, or how many UMIs we had for each individual gene. And then we also have, for each droplet, we detect the guide RNAs that were present. So we have a genetic perturbation associated with a gene expression profile. And the idea is from a very large pooled screen, you could then make these sorts of inferences about when I perturb gene A, here's 100 genes downstream that are dysregulated in their expression levels. So I mentioned previously that instead of just sort of this, conventional approach, we want to generate something called composite samples. So first we can just say mathematically, what is a composite sample? And the idea is that essentially it's relatively straightforward. Again, mathematically, it just represents a linear combination of outcomes. So we could think of composite sample number 1 as just being a weighted sum of outcomes of normal experiments. Okay, so like if sample I is a gene expression profile that results from perturbation to gene number 1, then, composite sample number 1 would just be a weighted sum of gene expression profiles that result from different genetic perturbations. And so it's not so complicated, just you could think of it either as a sum of expression profiles of different single cells or an average of expression profiles from different single cells. And the weights here can be, generally speaking, lots of different stuff. For us, they're just going to be either 0 or 1. Okay, so either a gene expression profile that was associated with certain perturbation was included in the composite sample, or it's not. But we don't have like 4.8 times, sample number 1 plus 3.2 times sample number 2, and no negative numbers or anything like that. So pretty straightforward. And then so again, this is like mathematically, we want to think about having generated composite samples like this. We could think of a second composite sample as just a different linear combination. If it's helpful to have like a visual, here's a composite slide. Okay, so literally we want to go do this, but like in cells, where you just have a bunch of stuff. Seems like it's a terrible idea, all sitting on top of each other. But if I gave you a talk that was formed in terms of composite slides, we would all be out of here watching the World Cup exponentially faster than we're actually going to be. So maybe that will give you some intuition about why we'd be interested about doing that in the lab. Here's a second composite slide, also features one of my dogs. All right, so how do we actually generate these composite samples in the lab in the context of PerturbSeq? We came up with 2 different methods to do this. And we ended up running kind of all of these experiments in parallel in the same pools of cells. So the first the first way of generating composite samples that we came up with involves something called cell pooling. So it starts the same. We have a guide RNA library. We introduce them at low MOI to cells. But now when we go to load the microfluidic device, we just put in way too many cells. Okay, so we ignore the instruction manual and we overload it. And what ends up happening is either the machine clogs and you wasted $1,000, or you get a actual usable sample out where you have a bunch of droplets that ended up with multiple cells per droplet. Okay, and so these are relatively straightforward to generate sort of random composite samples. The set of cells and their associated genetic perturbations that end up in any given droplet is just a random process. And what I read out from each droplet is now sort of an average of the RNA from all of the cells that were contained together in a given droplet. So in a droplet, all of the cells will be lysed, their RNA is spilled out, and we capture a sample of that RNA, and there's no weird interference or other nonlinearities. So it's just an average. And we, similar to the conventional screen, for each individual droplet, we now get sort of a composite gene expression profile. So how many molecules of RNA did we detect for each individual gene? And then we also detect which guide RNAs were present in each droplet. So we don't know which guide RNA went with which RNA molecule. At this point, we're not deconvolving to say these RNA molecules went from this cell and were associated with that guide, and those went from that. But that's not the mathematical setup. The mathematical setup is we want to generate these composite samples. Later, we're going to decompress them. The second method that we came up with for generating composite samples involves guide pooling. And now, again, we start with the same guide RNA library, but now we infect the cells at high MOI. So every cell ends up with multiple genes perturbed. And you can think about the transcriptional state of that cell as being sort of a composite outcome of each of the individual perturbations. a natural question is, what about all the interaction effects which exist in mythology and lore, and is unclear, how much evidence we have for. I'm not saying they don't exist. Surely interactions are more the rule than the exception. But so anyway, just to say that's a natural question of whether or not genetic interactions are going to confound this whole thing. We will test that by actually performing the experiment. And suffice to say, they don't systematically screw it up, which doesn't mean interactions between things don't exist. It just means that they aren't biased in one direction or another at like high frequency enough to screw up all of our results. All right, so that's the experimental setup. Once we've generated these composite data, again, the idea is somehow instead of like observing, individual cells with individual perturbations, we should be able to get away with a much smaller number of composite samples, and with the idea that the sort of outcome of each individual genetic perturbation on the expression of every individual gene possessing this kind of latent structure, which is just a matrix factorization, where in particular, the effect of each individual perturbation can be represented in terms of this sort of matrix factorization as, in particular, where instead of trying to directly infer the effect of a perturbation on the expression of each one of 20,000 different genes, what we're going to do, and the whole thing that enables this to work, is infer the effect of each perturbation on an entire sort of co-regulated module of genes. And yep, so that's what we do. We adapted an algorithm and came up with something called FRperturb, for factorize, recover, perturb. I'm going to go through this rather quickly, but if anyone wants to talk more about it later, that's okay. Essentially, we have this observed matrix in every droplet of the sort of gene expression that was observed in that droplet. And the first thing that we do is we factorize that matrix into sort of a droplets by modules matrix and a modules by expressed genes matrix. That's step one. Take your observed data, factorize it into U tilde W. Now we do this sparse recovery, where we take the sort of left matrix here, U tilde, and we have this matrix D, which is which perturbations were present in every droplet. And we just do basically a sparse regression to recover this matrix U, which again is for every perturbation, what was the effect of that perturbation on each one of these latent modules, okay? Then if we want to recover the sort of normal estimated effect size matrix, which is what's the effect of every perturbation on every expressed gene in the genome, we just take this U matrix and multiply it by this W matrix. And that gives us the sort of normal outcome to like a perturb-seq experiment. We did this in the context of studying transcriptional regulation in an innate immune pathway, specifically macrophages responding, closing that, I suppose, macrophages responding to stimulus with LPS. So it's a very highly studied pathway. This is quite a bit larger screen than had been done previously. We picked 598 different genes to perturb, which came from a variety of sources, including sort of a curated list of genes that are implicated through GWAS studies of IBD or Mendelian disorders, and a couple of other sources. And here's sort of the overview of the experimental design with this macrophage cell line. And we have kind of two arms of the experimental work that we did that are related to these two different methods of generating composite samples. So one of them is going to test cell pooling. The other one is going to test guide pooling. In each case, we ran a sort of very large conventional screen to generate validation data. And then in parallel ran sort of a compressed screen, either overloading cells in droplets to generate cell pooled samples, or in this case, overloading guides in cells to generate guide pooled composite samples. The other thing is that in this branch of the experiment, we did it with Cas9 sort of knockout of individual genes. And in this branch of the experiment, we did it with dCas9 crab knockdown or CRISPR-I inhibition of individual genes for a couple of reasons. One, it's interesting to just, because we're generating these big validation data, we can ask how concordant even the conventional branches of this screen are. Two, in this high MOI, where we're loading in multiple perturbations per cell, many cell lines are sensitive to having lots of double-stranded breaks. And so with the knockout version, you might get cellular toxicity at high MOI. Three, we wanted to run these two different methods anyway, but the sort of the guide pooling also potentially gives us the ability downstream to look at genetic interaction effects, again, which might confound us and which we might or might not be powered to learn in addition to each of sort of the first order effects. Okay, so we ran all sort of 4 of those branches of the experiment. In this sort of conventional knockout branch here, we did 19 channels of 10x sequencing. And in the cell pooled analog, we ran a single channel of cell pooled. So the sort of natural experimental unit to assess efficiency here is in terms of channels. Here we ran, I don't know, something like 90,000 cells compared with something like 8,000 or 9,000 cells that we ended up analyzing here. And then we, do a bunch of analysis to check for, do we learn the same effects in the compressed screen as the conventional screen? And then we can do a variety of downsampling stuff to say how many, sort of compressed experiment dollars give you the same level of accuracy as conventional experiment dollars. Okay, so first let's start evaluating the cell pooling results. As I said, 19 channels in the conventional arm versus 1 channel overloaded in the cell pooled arm. Notably, when we do this cell pooling, not only does every droplet end up with potentially multiple cells per droplet, but just due to the nature of the system, you also get a lot more droplets that are non-empty out of a single channel. This turns out to be a large source of the efficiency gains in this particular branch. But whatever, you pay for the same amount of reagents, whether or not you use them. So I'll take it. So we can compare first at a high level, sort of a picture of the regulatory effects that we learned. So I'm showing you here on the x-axis in each row of this heat map, we have a different perturbed gene. We're focusing on 50 of the genes from our set of 600 that had the strongest effects genome-wide. And every row in this heat map is a expressed gene in the genome that was regulated by lots of different perturbations. Okay, so this is just a global heat map of the sort of regulatory effects that we learned from the conventional data with 19 channels of 10x sequencing. Here's what we learned from a single channel of a cell pooled experiment. And just to give you an idea, some intuition before we get into the quantitative results, if we take a single channel of conventional data, it looks like crap. Okay, so it's not the case that this was just incredibly overpowered and we could have done 1 channel of the normal way. If you only use 1 channel of the normal way, you get incredibly noisy results. Here's some more quantitative analysis of that. First of all, we can look at a correlation in the effect sizes, where on the x-axis, I'm showing you effect sizes learned from our 1 cell pooled channel, where each dot represents the effect of perturbation to one of the 600 genes on the expression of any gene in the genome. And the y-axis shows you effect sizes that we learned from the 19 conventional channels. So they're pretty highly correlated, about 92%. Sign concordance is about 96%. We can then do these various downsampling experiments where we ask how many sort of conventional channels are needed to give you equivalent accuracy to 1 sort of cell pooled channel. And depending on the way that you slice and dice the data or what level or what sort of measure of accuracy you're interested in, we say we have between sort of a 4 and 20-fold reduced cost in the compressed arm of the screen without losing any accuracy. So you achieve equivalent accuracy. Yes. And I should say here that some of the gain is due to running the experiment differently with cell pooling, but also a lot of the gain here is due to our computational method, this FRperturb method, that does inference of perturbation effects at the level of these sort of co-regulated modules of genes. We can actually run that method on conventional data as well, and it turns out to be much better, I think, than existing methods, sort of gene by gene methods of determining regulatory effects. So I think I only have a supplemental slide on that, but just to say that, you know, if we took the conventional data, which is 19 channels, and we split it in two, so we have like a validation set, and then the other half we perform different levels of downsampling on, which is how we get to this result here. But we can run sort of existing computational methods on both of those sets of data and ask, are the effects that we learn between the sort of downsampled data set and the withheld validation data set consistent, just like this sort of scatter plot here. And with conventional sort of computational methods that do differential expression analysis or run an elastic net or something like that, they are like not concordant at all, at least compared to running the analysis with our method FRperturb. So that was maybe not the like clearest way of saying simply we have gains here because we ran the experiment differently and because we have a different computational method, and those two things both combined together to give us this sort of 4 to 20-fold improvement in efficiency. In the guide pooling branch of the experiment, we had about 66,000 droplets that we measure in the conventional branch. And we in total ended up with about 24,000 sort of guide pooled cells, but we're interested in a bunch of those ended up with actually only a small number of perturbations. So we're going to focus our analysis on cells that ended up with 3 or more guides. So the set of data that we're working with here are 8,400 droplets that ended up with 3 or more guides, and we're comparing them to this conventional data set with 66,000 droplets. So we can do kind of the same thing before, where we look at this heat map of effect sizes in the conventional data versus the guide pooled data with 8,400 cells. If you take 8,400 cells from the conventional experiment, you know, just sort of qualitatively, it looks much noisier. We can again do these sort of quantitative comparisons here. The correlation is slightly worse, but still reasonably good, I would say. And importantly, we can do these downsampling analysis, once again, to ask if you wanted to achieve equivalent accuracy in a conventional screen to what we get with our guide pooling screen, you need to spend about 10 times as many dollars. And that's combined between sequencing and library preparation. Okay, so we feel reasonably good about the method. I would note that this sort of second approach, even though the other method gets like up to 20 times better or whatever, and 20 is a bigger number than 10, we did some separate analysis and some simulations, and we feel reasonably confident, and in particular in our lab going forward, saying that guide pooling is kind of the more effective approach here. I mentioned earlier that one of the big sources of efficiency gains in the cell pooling experiment is that you simply get many more droplets out of a single channel. There's this kind of offsetting effect that effect sizes from multiple cells pooled together in a droplet are kind of diluting each other out. Whereas in the guide pooling, where you only have multiple perturbations inside a single cell, there's no dilution of effects. So we looked both in our actual data and kind of analyzed if you and in simulation, if you have cells with a single droplet or if you have droplets with a single cell or 2 cells or 4 cells or 8 cells, how does your validation accuracy change with increasing amounts of data? And here what we see in simulation, but also in the real data, is that actually the droplets with single cells in the cell pooling experiment give you more power, than the sort of droplets that ended up with multiple cells. Okay, so again, the efficiency gains there are coming in large part due to the fact that you just get a lot more droplets out of the reagents you already paid for when you overload the machine. On the other hand, with the guide pooling experiment, if you look at cells that have 4 or more guides, for the same amount of data, for the same number of cells, they have much higher validation accuracy or concordance with the withheld validation data set than cells that only received a single guide. And the same is true in sort of simulation experiments where in purple, cells with 8 guides, for a small number of cells with 8 guides, the correlation with validation data is much higher than it is using cells with a single guide. So we also tested this in a completely external to our study. We took data set generated by a different lab that happened to be massive and also very nicely annotated with cells that had a single guide or multiple guides. And we found the same sort of relationship where if you take cells from this external data set that they actually threw out, okay? So normally what happens with either multiplets or cells that receive multiple perturbations is those are just tossed out. So we took their tossed out cells, and if we only took their tossed out cells that had 3 guides per cell, again, for an equivalent number of cells, you get a much higher concordance with validation, withheld validation data, than the cells that they actually used in their analysis. And we were able to show that if they, you know, there's a sort of effective sample size that can be calculated, meaning, you know, well, essentially, if we take the data that they actually used, and then we include all of these data that they threw out, that include droplets or single cells with multiple perturbations, we can effectively double the size of their experiment. So they're throwing out all this data that they don't need to be throwing out. All right. Now, downstream of this, that's all kind of validation of this compressed method, both experimentally and computationally. Now going forward, we sort of have interrogation of the actual biological signal that we can pull out of these individual experiments. I've started to show you some of these heat maps already. Broadly speaking, we see that the perturbations can be kind of grouped into 3, what we can think of as co-functional perturbation modules. And we also find similarly 4 co-regulated expression modules. So these are sets of genes that kind of respond similarly to the broad panel of perturbation. And, you know, I'm not going to go through all the details, but if we interrogate, you know, sort of which genes, when perturbed, cause up or down regulation of these different co-regulated modules, for example, upregulation of an inflammation module expression or activation differentiation, an antiviral module, etc. Lots of that is consistent with what's already known in LPS signaling. We discovered a few new things as well. The other thing that we can do, as I said, in guide pooling, we can also potentially discover second-order genetic interactions. I would say that here it's important to note that we perturb 600 different genes overall, and so we are not at all powered to directly learn from that individual pairwise effects. The main reason being that the number of pairs of genes among a panel of 600 genes is 600 choose 2, which turns out to be a very large number. It's like 180,000 or something. So most pairs of genes we never actually observe in our data set, simply because there's so many of them. We didn't sequence 180,000 cells. So we absolutely do not have power at all. And this sort of illustrates the problem in general for studying combinatorial interactions, is that things grow as, like, you know, N choose O, if N is the number of genes and O is the order of interactions that you want to study. So it's an entirely intractable problem, I would say, to approach with sort of conventional experimental design. This relates to that comment that I had at the beginning of the talk, that we cannot hope for a better box to solve these combinatorial problems for us. This approach, however, has sort of exponential gains, potentially in experimental efficiency, which can kind of offset the exponential explosion in number of combinatorial interactions. So it's one way to make these sort of problems experimentally tractable. So anyway, we were not directly powered to learn individual pairwise effects. So instead, what we did is we tried to look for genetic interactions within modules, within like Go categories or modules as they were described in our data. We found some sort of intra-module interactions. So for example, if you look at TNF alpha signaling, we found some intra-module second-order genetic interactions, meaning if you take within that set of genes in Go term for TNF alpha signaling, we know that there are sort of second order interactions. So if you perturb two genes at the same time within that set, you have a surprising effect on the inflammatory program, I believe. And we found these sort of intra-module interactions for a few different modules listed here: antigen processing and regularization of chromosome organization, and we didn't find any sort of inter-module interactions. The only reason I say that, it's very sort of sparse and you know, it's not an entirely comprehensive result, but I happen to know that there are a couple other studies at the same time that found the same thing, where they find evidence of these intra-module interactions, and they find no evidence of these inter-module interactions. So doesn't mean that that's a rule of the universe, but there's at least evidence coming in. All right, so the next thing that we did was we asked, does the sort of, do the regulatory relationships that we learn from these genetic perturbations give us any insight into disease processes? With the important note that a lot of the genes that we're perturbing are under strong selective constraint. And so it's very difficult from GWAS studies to observe their effect or detect their disease relevance in the population. Because sort of large magnitude SNPs and so on are selected out of the population. There's some offsetting of that where things at lower frequency might also have higher effect, but suffice to say that it is generally harder to detect a lot of these strongly selected genes as being relevant for a particular disease through GWAS studies. So we hypothesize that if a gene in our panel is relevant for a disease, then disease heritability might be enriched near the downstream genes that a given perturbed gene regulates. Okay, so if gene A is somehow relevant to disease number 1, we might not see a lot of SNPs near gene A because they're selected out of the population. But we might, across the whole set of genes downstream of gene A, there might be SNPs that have weaker effects individually, and in aggregate, disease heritability is enriched across that set of SNPs near the downstream genes. So gene A regulates 100 genes downstream. Somehow this regulatory relationship is relevant for a disease or a trait of interest. We can't observe gene A's importance directly because it's under so much selection. But there are weak effect SNPs near all his downstream genes. And if we sum up all of those, you know, in an appropriate way, then we'll see heritability enrichment in ways that are otherwise difficult to detect. All right, so we did this sort of heritability enrichment of genes downstream of a particular perturbed gene, looking at 64 different traits, sort of, I think, not all of them, but many of them shown here. And what we found is sort of significant enrichment. So I've got individual perturbed genes here in each row and a lot of different traits for which we have GWAS summary statistics here in each column. And these dots kind of are indicating where there's significant enrichment for disease heritability in the genes that are downstream as determined in our perturb-seq studies from a particular perturbed gene. In particular, we find a big block of enrichment in both the knockout and the knockdown screens for a lot of sort of immune and blood composition type traits, which is good. This is, again, macrophages responding to LPS. We also wanted to know, sort of staying in the realm of population genetic type analysis, if the regulatory relationships that we're learning are consistent with what could be learned from eQTL studies. So there's kind of a thought in the field right now that cellular circuitry could be deciphered from running larger and larger eQTL studies. We'll see sort of SNPs near gene number 1, where the SNP is a cis EQTL for gene 1, and also a trans EQTL for gene 2. And from these sort of cis by trans relationships, through larger and larger EQTL studies, we might be able to decipher regulatory circuitry within individual cells. Okay, so that's a thought that's out there. People are really running very large EQTL studies, both with bulk sequencing and with single cell sequencing to see if this is true. So we wanted to know. We just did a bunch of genetic perturbations, and we learned through perturb-seq a lot of these regulatory relationships of gene 1 regulates the expression of gene 2. So we asked if there was significant overlap in the regulatory relationships that we learned in perturb-seq with cis by trans EQTLs that were inferred from a reasonably well-matched EQTL study that were primary monocytes stimulated with LPS. So we have a cell line of monocytes that we differentiate into macrophages stimulated with LPS, so pretty reasonably well-matched. Essentially what we found, which you may have already learned from reading the title of the slide, is there's no overlap. In fact, there is less overlap than you get with random pairs of genes. So it really does not seem to be the case, at least in this one individual example, that there is the expected consistency between these regulatory relationships that you learn in EQTL study and through direct genetic interventions. We do not see any enrichment from our knockdown screen, nor do we see enrichment from our knockout screen. In fact, compared to what we would expect with just random pairs of genes, we see a slight depletion. We can also say, if I look at curated pairs of transcription factors and their targets, are those enriched in the cis by trans EQTL data set? And they're not, they're also slightly depleted. On the other hand, if you look in the EQTL study and ask which pairs of genes are co-expressed, and then ask if those have significant enrichment in cis by trans EQTLs in the same data set, they're highly enriched. Okay, so they do various bits of pre-processing typically in these studies, like removing the top 20 PCs or something that's meant to remove kind of environmental confounders. But what we find is sort of consistent with the fact that most of the sort of cis by trans EQTLs still seem to be driven likely by environmental confounders. This is not proof of that. This is evidence that's consistent with that, and is my opinion of what is likely happening. You know, as to why this might be happening, again, there could just be environmental confounders that have not been accounted for. There's also this issue of sort of selective effect on all the downstream genes of each one of either the cis genes or the perturb genes. And in general, when there's selective effect on genes, either the cis gene or the trans gene, it's going to make them harder to detect in these population genetic studies. And sort of consistent with some of that, we see that the sort of downstream genes that we discover from our perturb-seq on average are in fact under greater selective constraint than your average gene, which might help explain why we don't discover them in EQTL studies. The same thing is true for curated transcription factor target pairs. On the other hand, this category of co-expressed genes that's very enriched in cis by trans EQTLs from this particular study have less selective constraint than the average gene, which one is free to interpret however they want, but you know, you might interpret that as being sort of less interesting and less likely disease relevant. Okay, that's about it for me. I would like to thank some of the people that did this work. As I mentioned, it went back quite a long time now. I'm at Boston University. Now this work started at the Broad Institute when I was in Aviv's lab, but especially much more recently, all the computational analysis has been taken on by Doug Yao, who's a Harvard grad student, has really done amazing work getting all of this to the end and over the finish line, and thought of many things that I would not have thought of. And Doug is also co-advised by Sasha Gusev over at Dana-Farber. I think that's it. Happy to take any questions. Yeah.
I was wondering, have you tried doing like using nonlinear methods to express the relationships between some of the elements? Because it's a linear regression, but I think some of them can be modeled nonlinearly.
Well, how would you do it? What, like what? Oh, yes. I was instructed to do that, and my response was, I will definitely forget to do that. So the question is, if we have tried any sort of nonlinear computational methods to sort of infer perturbation effects, and no, the answer is no, we have not done that, except for in the case where we're trying to infer these interaction effects. Okay, so that's somehow nonlinear. Otherwise, we have not directly, I mean, besides in sort of generalized linear regression senses. But yeah, I don't really know the way to do it. My favorite analogy there is, you know, linear and non-linear is like, it's not like apples and oranges, it's like bananas and non-bananas. Okay, so which non-banana do I want to use? I don't know, there's a whole universe out there of non-bananas. So that's my main problem, is I don't know what exactly to use. It's entirely possible something would give us extra power, but I don't know which class of function. Yeah. Oh, yeah. Oh, no. Ah, okay. Yep. Hmm.
So, yeah, I guess you describe it when you got the comparisons there, and how to break down what's in terms of like where the like efficiency improvement comes from in terms of like the sparse recovery versus the
Yep. So we can do a couple things. One is, let's focus on this red line. So here, as I said, what I did was I took the conventional data and I split it in two. I'm going to use 1/2 as the validation data. Then I'm going to downsample the other part, compute effect sizes, and ask how correlated they are with the validation data. Okay, so that's the y-axis in this plot here, is correlation with withheld validation data. The dotted line is the correlation between my cell pooled experiment and the withheld validation data. The red line is showing you if I analyze the conventional data with our FRperturb method, if I use a single channel of conventional data, what's my correlation with the validation data? If I use 2 channels and so on. Okay. Now that's using our inference method on the conventionally generated data. If we take sort of existing inference methods and run them on the conventional data, you get these blue and these green curves for 2 different methods. So from this, we can say that, like, you know, this dashed line here, one conventional data, sort of one cell pooled channel analyzed with FRperturb seems to be better than at least 10 conventionally generated data analyzed with conventional methods. So this basically, here we get some number of, if you use the same computational method, you get like a 5- to 6-fold improvement, meaning where does this red line intersect the dashed line? In some of these other, this is more like a sensitivity analysis. In this sort of like precision recall analysis, the answer is like it's maybe 4-fold better at least. Again, we can't really tell because these lines can only be downsampled so far. So then what we did is we took our cell pooled channel and we downsampled it on top of downsampling the conventional data until we made it as bad as these red and these blue lines. So once you do that and then account for differences in sequencing, we had to sequence extra here because there's so many more droplets for a single channel. You get to 20-fold. So we don't take the biggest number and just say it's 20 times better. We just give a range and say, in some analysis, it's like 4 times better. And, you know, the most generous analysis, it's 20 times better.
Versus like the differences between your like inference on top of it and the, I guess,
yeah.
Other methods.
Yeah, so again, if you analyze the conventional data with our method, which we think is better, then the compressed experimental workflow is like 5 to 6 times more efficient. Okay, and that's in this sort of correlation with withheld validation data set. If you instead ask sort of a recovery question, so this is like, take the significant effects as determined in the compressed experiment or in the downsampled conventional experiment, and ask how well they correlate with those same effects learned in validation data. Instead, we could ask, you know, do we recover the real effects that we learn in the validation data, and how many sort of conventional channels do you need to have the same precision recall as you get with the sort of compressed channel? And so again, you can slice and dice different ways, use different computational methods, and look at sensitivity or specificity. And so we just end up with this range kind of. Yeah. Yeah. Yep. So yeah, so a sort of nice feature of these experiments is that we need to know that the structure generally exists, but we don't need to know any of the details. So pretty much you need to know the story is true, like things are generally organized into co-functional sets of genes and co-regulated sets of genes, and that you could represent this sort of high dimensional thing in this factorized way with some sparse parameters. But you don't need to know any of the details of which genes are co-functional and which perturb. So in that sense, we don't need like training data that we need to behave as the same as test data. However, if you have like different cell types in the sort of same experiment, then you'd probably want to treat those differently, you know, or at least that would be my initial inclination. Here we have one cell type, everything behaves the same. So like I have one set of co-functional modules and I'm applying those to all droplets or whatever. If I had different cell types that I wanted to run in the same experiment where I thought the effect of a perturbation in one cell type would be different from another cell type, out of the box, I wouldn't just do this. You know, I would want to somehow make note of the fact that perturbations might do different things in different cell types. Yeah. We found pretty robust performance picking kind of a wide range of parameters from like 10 up to 100 or something. So you definitely don't need to like nail it. You can do, and what we did is just some like cross-validation analysis to see, again, we did a whole lot of these. Even if you only ever generate compressed data, you could split it in half and then ask, you know, which value of R, the rank or the number of modules, gives you the best consistency between the two. So so you don't need to like nail it. And if you want, you can do these sort of data splitting and consistency checks. But in particular, you know, I'm very interested, as I mentioned in lunch with some of the students, in, you know, what is here, this is entirely statistical manifestation of something that's actually interesting. Okay, that's how I would put it. There's some actual interesting biology, which we think of very loosely as a cellular pathway, and there's different cellular pathways and things are organized in there. The fact that stuff operates within cellular pathways manifests statistically in these sort of patterns in matrix factorization and so on that we can exploit with these experimental designs and these algorithms. But like directly, I don't know how to interpret the rank, you know, in terms of like a profound parameter of the organization of the cell, or like the number of pathways that exist in the cell, just as I don't know how to interpret any one of those columns or any one of those modules. And that's not unique to this. I have that general opinion about every single transcriptomic or genetic study that exists anywhere. It doesn't stop people ever from just interpreting them, you know, at will. But, you know, I don't know if any of that means anything. So here it's cool. I get to be agnostic because, again, I just care about the statistical properties of the patterns and the structure in the data and the fact that I can exploit those through experimental design. And I haven't really said anything about the biological meaning of the value of R or each individual column or anything like that. But that's the actual interesting question. Yeah. Yeah, I do. We did that here, yeah, not because we know like the value of R or anything like that, but because, as you said, it's well studied. So like if we see Mighty 88 either does or does not have an effect on the inflammatory response, we know the answer. And so it's good when developing wacky technologies to like have that baseline. Yeah. Okay. Oh,
How do you pick which perturbations to make? Like, is there a way to do it like theoretically, would you say if these are co-regulated, these would be my most informative perturbations?
well, so there's, I guess, two parts to the question. One is like, we have this panel of 600 genes and there's what goes in that panel. And then there's, and then there's a second question, which may not have been what you were asking, which is like, which perturbations go together in a composite sample? The answer to that second question is the fact that the mathematics work where those are entirely randomly chosen, we take advantage of through the physical process of stuff gets loaded into a machine and randomly ends up in a droplet together or not. And again, we know how the math behaves, so that's okay that they're randomly generated. We don't actually have a way in high throughput to pick exactly which stuff goes together. It's much easier to just let the sort of stochastic process play out. There's another reason that I sort of prefer them to be random, which is, first of all, again, there's like good mathematical understanding of why kind of random features work, very much related to like why doing randomized clinical trials are a good idea instead of trying to exactly decide who goes in every group. One of the reasons is that, okay, so why do we randomize clinical trials? There could be unknown confounders. And if we randomize, it's very unlikely that we segregate people into treatment and control groups in a way that is directly aligned with some confounder. So the same thing is true here. If you try and use what you think is exquisite knowledge of stuff that might be co-functional, or you want to construct your composite samples in one way or another, that could be good, but you also run the risk of exactly aligning yourself with a technical confounder or a batch effect or the exact last thing you wanted to align your experiment with. And if you design it randomly, you essentially do that with 0% probability. The other question of which genes go into the panel of 600 here. So yeah, we chose them from stuff we think we know should be involved, things that are relevant through GWAS studies for diseases that likely involve this pathway, and same thing for sort of Mendelian disorders. And we also use some data from some previous screens.